Hi, welcome to the webinar. My name is Hojun Song. I am the PI of the NEON Biorepository. NEON Biorepository is a long-term, 30-year community resource optimized for monitoring and forecasting ecological responses to environmental changes at a continental scale. It's a physical infrastructure associated with the NEON, and our core responsibility is to receive samples from the field sites and accessioning it, curating it, vouchering those samples. And then we strive for long-term preservation, and then we respond to external research communities through loans and data accessibility. We are physically located at the Arizona State University campus in Tempe, Arizona, within the ASU bio collections. And the NEON Biorepository currently includes myself as a PI, three full-time collections managers, one full-time sample preparator, and then three full-time bioinformaticians. So it's been growing, and it's been growing since 2019, and we project that we're going to grow until 2049. So you hear a lot more about our facility as well as the samples that we have, as well as the data portal. So I'll pass on to Chandra and take it from there.
Yeah, thanks, Hojun. That's really great. I am going to go ahead and share my screen then. Ta-ta, and move you guys back over here. Okay, so hi everyone. My name is Chandra Earl, and I am a biodiversity informatician with the NEON Biorepository. And so today I get to provide an orientation to the NEON Biorepository and show you all how to search for and request samples from us through the Biorepository portal on the neonscience.org website. So briefly, just kind of real quick, here's the agenda for this webinar. So first we'll go over just some general background and information for the NEON Biorepository, like what we are, what we do exactly, and what our holdings contain. Next, we'll go over just some ways to interact with us, including how to use the portal and what that typical loans process looks like. And then lastly, if we have some time, we'll go over how NEON samples are currently being used in research. So first, and just basic question, what is the NEON Biorepository? So Hojun gave an excellent sort of overview into what it is, but just to sort of reiterate, NEON collects data and samples via three collection methods at each of the 81 field sites, right? So we've got the automated instrument systems, we've got observational sampling, and we also have airborne remote sensing. So the observational sampling is what we're going to focus on, and that encompasses both the observations and the physical samples that are collected by people in the field. So most specimens and samples make their way to the NEON Biorepository. So now what makes the NEON Biorepository very unique when compared to other specimen collection facilities is one, that sort of deliberate ecological interconnectedness of our samples designed to monitor ecological change. And second, unlike most, we have a primary objective to make those specimens available for consumptive research. So we do really encourage people to use and consume our samples. Physically, the NEON Biorepository is co-located with the Arizona State University Natural History Collections in Tempe, Arizona. So it's about 2 miles southwest of ASU's main campus in what is called the Alameda Building. Currently, in 2026, this is sort of what the inside looks like. And so you can see that, of course, since most of this space is owned by ASU, most of the space is physically shared with the Arizona State Natural History Collections in blue here. There are also classrooms and offices that are used, of course, and there are a few NEON Biorepository specific rooms in the mix here in orange. But one part of this that is specifically unique to NEON are the cryo collections. So those are those specimens that are kept in the -80-degree freezers and in liquid nitrogen. That area that I just showed is actually only a third of that entire Alameda building, so this blue part down here. The first phase of the NEON renovation for the biorepository finished in 2020 and included renovating the cryo collection space out here in Orange. And so this is what that sort of looks like now. So we've gone ahead and put in those freezers, the liquid nitrogen tanks, and a processing space for incoming frozen specimens. We are currently undergoing our second renovation to expand into that second third of the building here in yellow. And so this space is specifically designed to hold the next 25 years of sample collection, and it is expected to be finished in 2027. So we're really excited about this work. So what exactly does the NEON Biorepository do then? Well, the best way to think about the NEON Biorepository is to think of it like a turntable for samples, where samples and data are ingested, they're assimilated, and then redistributed back out to the community. So starting at the top, physical collections. Physical samples are collected at all 81 field sites, of course. But the NEON field sites are distributed across those 20 ecoclimatic domains that represent the distinct ecological regions. So each domain has a field office or a domain facility designed to oversee each of those sites within their purview. So when those samples are collected, they are first sent to that domain facility. And so usually some sort of processing happens here, like subsampling or any downstream preparation that needs to happen. But from the domain facility, samples are either retained for teaching collections, they can be sent to external contractors, or they can be sent directly to the biorepository, just depending on the specific sample type. We, of course, then receive samples sent directly from the domain facilities, but also from external contractors. But that also, again, depends on the sample type and the analyses being done on all of those. What happens to a sample once it actually reaches us, really, again, of course, depends on the type of sample that it is. Most stuff we get sort of just involves your typical curation of those samples, so putting them away where they need to go. So typically that is organized by the type of sample, what site it was collected at, the year and taxa if appropriate. So for example, you can see this is a Laura Steger, environmental and zoological collection manager, putting away mammal skins. This is Azar Hussein, who is our cryo collections manager, putting away samples into liquid nitrogen storage. And then these are just some examples of how those samples are organized in the collection, right? So these are some pinned mosquitoes. These are those -80 freezers that we saw earlier. And then this is an example of what some alcohol collections look like. On the other hand, some samples that come to us need to be prepared before they can be put into long-term storage. So herbarium specimens need to get pressed flat and glued onto herbarium paper. We do make little envelopes for dried algae, mosses, and lichen samples that are too small to be put onto that paper. And we do have to rehouse the herps and the fish into appropriate jars for storage, for that long-term storage. But perhaps the most time-consuming but rewarding prep involves the small mammals. So these typically come to us frozen, and most are prepared as skins. So we take them and we stuff them with cotton. But all of those internal samples are taken out and sampled and frozen for anyone to use. And whole skeletons or skulls can also be prepped using the dermestid colony. So the dermestid colony are those flesh-eating beetles that clean tissue off of bones. And so you can see a picture of them here where all of those little black dots are the tiny beetles. And this is a photo of someone prepping one of those small mammal specimens. We do also measure various traits for the mammal specimens, and that includes your body length, your tail length, ear length, body mass, reproductive condition, etc. That is all also available for each of these specimens. But because this part specifically takes a lot of work, we do have a full-time mammal sample preparator on staff named Jaime Galvez, and Jaime is fantastic in not only prepping the specimens, but also helping us train other students to do this work as well. We also image a good bit of our zoological collection. So this is a photo of the imaging station. Each beetle and mosquito species gets at least one image, and every single fish, herp, mammal, and herbarium specimen gets imaged as well. And so as you can imagine, since we are continually getting specimens in, this is an ongoing process, and so we'll never be at 100% coverage for all of these, but we do our best to keep up. And again, all of these images are also available for researchers as well. So now what exactly do we have? We've kind of touched on a little bit of the holdings that we contain, but what exactly is going on here? So like I said earlier, one of the things that makes the NEON Biorepository unique is that deliberate interconnectedness of our samples. Part of which comes from the fact that we not only heavily analyze and process those samples that we collect, but also that we store generated samples from across that entire protocol, right? So this processing happens at any stage, whether it's in the field, at the domain facilities, at the external contractors, or even at the biorepository itself. So this is a list of all of the NEON protocols that result in samples that come to the biorepository. And all of this sort of means that ultimately the protocols and that the resulting sample types that we store are not a one-to-one thing. So for example, in the case of these aquatic protocols, we're not just going out and sort of scooping things out of the water and putting them on a shelf for people to use, but we're also analyzing a lot of those samples to create data to publish to the data portal, and also more sample types that get stored in the process of analyzing those samples originally. Now, if you're familiar with our data already, you sort of already know this, but our processes can be very, very complicated. So for example, this diagram on the right shows our ground beetle pitfall trapping protocol and how it results in 10 different sample types that have to be stored separately, just depending on how the samples are processed. So basically, we put 3 cups of fluid in the ground and check them every 2 weeks. The collected stuff out of these containers can either be combined together or kept separately, just depending on how much was caught in each of these cups. But ultimately, these are sorted into beetles, invertebrates, small mammals, and herps. So sometimes individual beetles are also taken out and they get DNA sequenced or barcoded. So we do also get the pinned beetles from that and the resulting DNA extracts from that process as well. But ultimately, you can sort of see how the processing of these catches results in many different types of samples that we store at the biorepository. And on the left here, you can see some of the protocols that we have related to the number of sample types that we get from each of the protocols. So you can see that, again, this is not a one-to-one relationship, that we do tend to get a lot more than sort of your normal collecting protocol experience. We currently have about 715 samples available, and we do ingest on average 100,000 specimens a year. So our annual sample intake, when divided sort of vaguely into taxon and material types, looks something like this. The purple bars here represent vouchers, or those individually prepped and stored specimens. So this includes, for the plants, your pressed plant specimens. For the invertebrates, this includes pinned beetles and mosquitoes. And it also includes the individual, those small mammal vouchers that we create as well. The pinned invertebrates are by far the largest intake that we get here, of course. The second largest sample type is a bulk sample type, so here in orange. So this sort of means that the samples have not been separated out into individuals, although there might have been subsets taken out and analyzed from each of these. And so this includes the environmental samples that we get, so the soils, the precipitation, those dust samples. But the second largest intake is primarily due to the soil microbe samples that we receive. But this also includes the surface water microbes and the benthic water microbes as well. For plants, this typically is represented as collected macro and microalgae samples that are stored in glutaraldehyde. And for the invertebrates, this mostly includes those bulk beetle and bulk invertebrate samples from earlier that we talked about from that ground beetle sampling. This blue bar here the blue bars represent any tissues that are collected. So for plants, this mainly represents frozen leaf tissue that is meant for genetic archive. But in the vertebrates, which represents that third largest group, this is from the small mammal tissues that get sent to us. Since the small mammal protocol, the vast majority of the mammals that we collect are actually released. They are not sent to us at the biorepository, but we do collect tissues from those that are released. So we'll collect tissues like hair, feces, ear punches, and blood. And so those are all collected and sent to us and frozen as well. And lastly, if you'll sort of remember from earlier, we also get sent DNA extracts from external contractors where there is sequencing involved in that protocol. So that's this green color here. And so we get DNA extracts from things like microbial PCR analyses, invertebrate and vertebrate barcode analyses, and also genomic extracts from pathogen testing of the mosquitoes, ticks, and small mammals as well. So again, all of those come and are stored at the biorepository. So how does someone go about then interacting with the biorepository? We've talked a lot about what we have. How do I get my hands on some of these samples then? Well, there's a lot of different ways to interact with us, both virtual and in person, if you ever happen to be in Arizona and would like to visit. Of course, the primary digital way of doing so is through the NEON Biorepository sample portal. And so this is essentially our centralized database that we used for tracking specimen ingests, tracking loans that go out to researchers, and let folks search our holdings for available samples. And so this portal is currently managed by 3 people. So we've got Ed Gilbert, who is our Symbiota liaison. So Symbiota is the underlying code base that we use to run our portal. We've got Kelsey Ewell, who is our data science specialist, who analyzes and tracks the correctness and the longevity of the data in the portal itself. And then we've got myself, who primarily manages the NEON-specific code developments and integrations with the main NEON site. So we've put a lot of work over the past couple of years integrating the biorepository sample portal into the main NEON website, mostly to more align messaging and make it easier for users to move between various parts of the site. And so all of the information that I'll be going over in this next little bit lives under the samples and specimens tab of the NEON page. And so this, of course, includes the portal itself, pages about the samples, extra resources for finding and accessing sample data, and information on how to request and use those samples. For most people, this process sort of looks something like this. Where first we're having to browse and figure out what might be useful for your research. We might then search for those samples, and then lastly, we need to go about requesting those samples. So first, how does one go about figuring out what it is that we even have? Well, the primary way is via the sample type browser. And so this is available under the About Samples tab, see sample types, and it looks something like this. So this contains all of the sample types available at the biorepository, but we've organized it in 3 different ways. So we've got this organized taxonomically in case you would like to see what sample types are available for a specific taxonomic group. We've also got it organized by protocol, so you can see what sample types are the result of any one of our protocols. And then we also have a preservation method, so you might be interested in seeing what sample types are frozen sample types, or what sample types are those genomic sample types, and you can explore it through that as well. So this is what one of those sort of drop downs looks like. And to be fair, it can be kind of difficult to understand what each sample type means unless you are very familiar with that protocol. So I do always recommend using that documentation to get a sense of what you might be looking at. So for someone who doesn't understand our ground beetle protocol, might not understand what these sample types mean. But again, sort of bringing back that ground beetle protocol here, we can see pretty easily how each of these map to each of our sample types, right? So we've got the DNA extracts, we've got the pinned specimens, We have the beetles that are from pooled catches. We've got beetles that are from pooled catches, and then same thing with the invertebrate bycatch. We've got invertebrate bycatch from pooled catches and invertebrate bycatch from unpooled catches as well. So clicking on each of those sample types brings you to its sample type profile. And so this is a page. So this is actually just one page. I've split it up into two to fit on the screen. But it gives you more information on that specific sample type. So this is the page for the beetle DNA extracts, and you can see that there is a section, there's an about section, which just sort of talks about what this sample type is, how it's collected, how it, sort of where it sort of comes from in the protocol, how these are stored. There's a statistics section. So there's 21,000, we've got 21,000 samples for this specific sample type across 667 species. We've got the citation information, which gives you just the citation that you need to use in case you're using any data from this sample type. And of course, any of the linked NEON data products and also the protocols from where this sample type came from in case you need to explore or link out to any of those as well. So one thing to be aware of is that while the NEON Biorepository is the primary source of samples for the NEON project, some samples and specimens are stored elsewhere. And you can look and see what those are again, under the About Samples tab in the Sample Repositories page. So most of these are due to specimens being collected before the biorepository was built out, but some of them are also due to regulatory laws and requirements of specific locations that we collect at. And so you can get a sense of what those might be in this page. So for example, all ticks that were collected prior to 2024 were sent to the U.S. National Tick Collection at GSU. And so, but from 2024 on, these tick collections are sent to us at the NEON Biorepository. But anyone who wants to get physical ticks from earlier dates needs to contact GSU because we do not have those samples. So the second step in this process, or maybe an additional step, if you will, might be searching for those specimens that you want. And so the primary way to do that, of course, is through the Biorepository sample portal. And it looks something like this when you click on it. It's just a pretty basic search form with various sections. And I think these are pretty self-explanatory, but I'm just going to go through them real quick just so you can see at least what they look like and how to use it. So the primary thing that you do need to do when searching for samples is to designate a specific sample type. And so this will literally not let you search unless you pick a sample type. So clicking on this sort of all NEON sample types at the biorepository will bring up something like this. And all these tabs and all these dropdowns are the exact same tabs and dropdowns that we saw earlier in the sample type browser. So you can pretty easily click through and figure out what it is that you want, which sample types you're interested in, and move on from there. While we've also got this second section, while clicking on this all NEON sample types at other repositories brings up all those other repositories that we discussed earlier. So for example, you'd still have to contact GSU to loan out a tick specimen from prior to 2024, but you can get kind of a sense of what it is that they have by clicking this sample type and searching in their holdings. The next section lets you select specimens by domains, sites, state, plot, whatever county, elevation that you might be interested in if you need something from a specific location. There is, of course, a date section for specific collecting seasons or months. And then we also have sample properties, which is kind of like the miscellaneous category, but it also lets you search for samples with specific identifiers if you do happen to have that information. And so going into what each of the identifiers are for our samples is out of scope for this particular talk, but each specimen and sample can have multiple identifiers, and if you happen to find one of those in the wild, or if you know what it is that you specifically want, you can search for it within the identifiers. You can also filter out any specimens that you're interested in using the checkboxes. So specimens with images, you might want specimens with published genetic data, or you might want specimens that are available for loan, because we do keep records from all of the samples that we get, including those that have been constructively consumed or destructively consumed. And then lastly, if you do happen to be looking for a specific taxa across any sample type, you can do that as well. So this is what the search results look like. So for example, I did a search looking for salmonids across all fish sample types. So in this case, we do have two fish sample types. We've got the fish specimens themselves, and we have DNA extracts from those fish. I searched for domain 16 and 17, which is the Pacific Northwest and the Pacific Southwest. And I also only wanted samples that are available for loan in case I wanted to request any of these for loan. So once you do a search, like I said, it'll look something like this with all of the records sort of laid out. So we've got 151 of these records available. But you can see an overview of each of these records is visible. But there are also different tabs that you can click through at the top. So you can see the list of species from this search. So the search that I did was a whole family. These are just the individual species that come up with this. You can see a map of the collection sites or plots for each of these records. And then you can see any of the images available for these records, again, if there are any. Clicking on a full record brings you to an individual record page. So there is a lot of information on each of these pages. And so this includes that identifier information like we talked about earlier. It also includes some taxonomic information, so the taxon, the family, the determiner who determined that specific name. It includes collection information, so who collected it, the date it was collected, the location, specific location where it was collected, including the latitude, longitude, and elevation. It also contains any just extra specimen details that we might have added to that record. So this specific one, has a description of sort of what this thing is, the life stage, how many, there's obviously one fish specimen, how it's prepared, and any notes available with it as well. It also contains any linked specimen records or any linked specimen images that we have or may have taken with it. And it also contains these associations. So associations are linked specimen records. So for example, this, of course, is a record of this fish, but we also do have the DNA extract of this fish available as well, in case you wanted to click on and look at that record. You can see that it is linked. So similar to the search results, there are extra tabs at the top, again, depending on the information that is available for the specimen. So you can see this one has a map of where it was collected and also a link to its DNA sequence in the Barcode of Life data systems. Of course, just sort of scrolling through records might only be helpful for a little bit, but you can also download those records and search through any of those fields in mass if you wanted to. And so that's available in this button here on the top right. And once you click it, a little pop-up will open looking up something like this. You'll click download and it'll download just a zip file of the data that you were just looking at. So if you are familiar, the data do get downloaded according to the Darwin Core standard, which is maintained by TADWIG. And this is this is because, of course, it is a standard that we like to adhere to, but it also means you can sort of easily combine that data with other sources that are in Darwin Core if you really, really wanted to. Outside of just directly searching for samples within the sample portal, you can also discover samples through our species checklists as well. And so we provide these checklists for each site and domain across these various taxonomic groups. So for example, if someone wanted to get an idea of what herps have been collected at the Harvard site, you could pretty easily do that. One thing to note here is that the underlying data are only what has been collected at a site. So these lists are not complete or authoritative, but it can be a nice tool to get an idea of what's in an area for a taxonomic group. And so you can easily get at what samples are underlying this data by clicking on any of the magnifying glasses next to any of the names or all specimens. So for example, if I wanted to see what specimens, I wanted to see what these are a type of salamander, what type of, what's it, Ambystomia today, are at this site, I could click on the magnifying glass and the same exact record search would pop up, except I'm looking, of course, for Ambystomia today across Harvard. So I can see what specimens, again, make up that taxa. I did wanted to walk through some special considerations when searching for samples that people might not realize, or at least these are all like reasons why you might not get the samples that you expect in your search, or at least they're reasons why, like, I've done a search and like the specimens that I was expecting have not popped up. So first, we've already talked about this, but like we don't receive all resulting samples from a protocol, right? Of course, some are sent to a different storage facility, some are retained by the external contractor, and some are retained by the domain facility. So it's just a good thing to know that we don't necessarily always have them. Someone else might have them. Second, it can take years to receive certain sample types. So I've definitely done a search where I've expected something to show up and I realize, oh, we haven't received these samples yet. And this is usually because samples are often sent to us in bulk. So they're not, sometimes, again, depending on sample type, they're not always just sent to us little by little as they're receive and ship them out. They will hold on to them, wait for them to sort of accumulate, and then send them in bulk. So for example, we just curated the pinned beetle specimens from 2024. We just got those a couple months ago. But you can see that it takes it can take a few years to get all of that information into the biorepository, but also the biorepository portal as well. Some specimens are only sent to us by external contractors at the end of their contracting period, and those can also be years long, just depending on what the contracting period is. So we do have to wait for those to come to us. Third, some sample types only get archived if there's material remaining after analysis is done. So for example, the root cores. So, people go out and they will core roots out of the earth. They'll dry them, they'll oven dry them, grind them up, sort them into different sizes, and then they'll do various analyses on them. But we only get samples if there's material left after that analysis. So sometimes you can expect there to be a sample that doesn't actually exist because that it has all been used up in whatever analysis. Sometimes, and I think this one can get pretty confusing for people, is that intermediate sample types are also often given IDs or barcodes and recorded in NEON data. So you might have a download of a NEON data product and see a field that's just called sample ID and might think, oh, that sample ID must be available in the biorepository, which isn't necessarily true because it's an intermediate sample type. It's great for connecting, the different various tables across that protocol, but because that sample doesn't exist anymore, of course, it was just an intermediary to get to the final one, we wouldn't necessarily have that ID in, you try to search for that ID and it won't pop up. Almost done, I promise. Collection protocols can change, of course. So this can change anything from the number of samples collected to how identifiers are structured. So, you might be expecting, 3 sample types per year per site for this specific sample type, when in reality, it's only 2 or something like that because the collection protocol has changed in that period of time. And then lastly, collection goals, again, vary across protocols. So we're not going out necessarily collecting everything that is at a site, right? Vertebrates, so those small mammals, those herps and those fish, are only collected, we only collect the accidental mortalities of those specimens, right? Most of those are labeled as occurrences. And then the plants are only collected really opportunistically for taxonomic or genetic archive, right? They're not meant to be a sort of change over time collection. So we just sort of might have whatever the field site deems is good to store for archive for the plants. But these, that sort of like expectation can get a little confusing, I think, to people sometimes. And then lastly, how do I go about then requesting one of these samples? Okay. Of course, we do send out loans to anyone who is interested. Funding is not required unless it is an unusually large or complicated request. Since every loan request is different and NEON is pretty complicated, we do work pretty closely with the researcher to make sure that they understand what it is that we have and that our samples are ultimately going to be useful for them. So first, of course, all loans start out with a request. This can be through direct contact for us, that happens quite a bit, or it can be through the sample request form. This form is available in the Loans and Archival Request page under the Requesting and Using Samples tab. And so that just pops you up to a page that looks like this, and the button for that form is down at the bottom here. So the form does look something like this now, with it requiring some information up front, like contact information, funding or grant information if that's applicable to you, what samples, of course, you want, and what you expect to use those samples for, or any expected products, that you expect to get from your analysis. We are aware that this can be a lot of information up front to someone who might not have it yet. And so we are actually currently in the process of revising this form and just making this process a bit easier to manage. That being said, of course, you can always just directly contact us to talk to us first before filling this out too. That's totally acceptable and happens all the time. The sort of second step here is mostly a review step. So this is mostly in cases where there are multiple people asking for a specimen, or if someone wants to destructively sample a large portion of a sample type. We do also mutually develop a sample use agreement, which is a sort of signed contract with a researcher during this step as well. So the exact contents of a sample use agreement will, of course, vary across every single loan and every single request, of course. But the general information that it includes a list of those approved samples, the scope of that research, the loan terms, any handling or data publishing requirements that we have, any citations, and of course any special provisions that need to be worked out with a researcher. We'll do that as well. But the most important part to note here is that this is something that is developed mutually with the researcher themselves, so that everyone is happy at the end of the day. It just sort of lays out what the expectations are, I guess. And so once that is sort of signed and good to go, we pull the samples, pack them up, and ship them out. So we do require a signed receipt upon someone receiving those samples, and this is just to sort of officially say that yes, you've received those samples. They're now under your care, your responsibility, until if they are sent back to us at the end of the loan period. So these are sort of the current stats for loans, if you will. So half of the inquiries that we get result in loans. And we currently have 77 active loans across 140 samples and 44 sample types. Currently, our most popular sample type, if you go by the number of samples that are loaned out, is of course the pinned beetles, followed by the bulk invertebrates and alcohol, then the prepared mammals, the bulk beetles and alcohol, and fifth is mammal feces. We, of course, are also a physical location as well. And so this means we do welcome visits and tours, and we do have some hosting capabilities, just depending on what your event is. And so here you can see Kelsey giving a presentation to the 2025 NEON RCN workshop group. We definitely, of course, also welcome visiting researchers and are happy to give anyone a tour. And so this is Laura posing with some visiting researchers that we had last year. This space is not open to the public, though, so you do have to contact us beforehand to set something up. So just as an FYI. And lastly, we are pretty involved in the community and do our own research when we have the time. And so you'll often see us at conferences or at outreach events talking about the biorepository. So this is Issa Bentoncourt, who is our entomology collections manager, giving a presentation on the beetle collection. And here's her again doing some tabling at a National Geographic Live event. This is one of my students giving a poster presentation about some research that she did with the bird data. And this is our PI, Hojun Song, who you met at the beginning of this, dressed as a grasshopper, giving a presentation about the NEON Biorepository at a Halloween-themed event that happened last year. And so lastly, sort of how are neon samples being used then? So how are they being used in research? And we've got some pretty cool use cases that I'm hoping we have time to get through. So our primary goal, of course, is to get people to actually use the samples, not necessarily to store them long term. We want people to destructively use them in research, and we're hoping that that's what people do. Just like our loans and inquiries, research using NEON data and specimens is sharply climbing. So this is a graph of the publications and citations across the entire NEON ecosystem. But this is a Google Scholar that sort of is tracking just the NEON Biorepository, either specific research or anytime someone mentions us or anytime someone cites us, something like that. And again, you can see that the citations for this is climbing pretty dramatically, which is very exciting for us. Because we do want people to use the samples and get them out into research. So last year I took a look at sort of all of these published papers and looked at also the loan requests that we came up with and that we've received, and came up with sort of 8 different broad research topics, if you will, that folks are using our samples for. And so the next few slides are just published use cases of each of these topics. First and most common is the biodiversity and taxonomy topic. And so this specific researcher used our bulk invertebrate samples in alcohol to search for, so they used the samples from Puerto Rico to search for and describe a parasitic wasp. And so we've had a few of these papers come out, but this one is particularly of note because they named the species after the NEON program and specifically mention how ecological monitoring from the NEON program resulted in a benefit to another discipline. So in this case, taxonomy. A use case example for studies that use DNA or RNA to characterize species include this one that took our frozen tissue from small mammal ears, small mammal ear punches. They sequenced them and then used the results to sort of characterize these small mammals into phylogeographic lineages, which was pretty cool. We do actually get quite a bit of disease ecology research being done with the samples, which makes sense given NEON's focus on collecting pathogen data. And so mosquitoes, for this specific one, are collected in bulk traps and sorted out to species and life stage. Some can be pinned as shown here, but most are stored in bulk specimens in alcohol. And so these researchers took those bulk mosquito specimens, ground them up, and characterized the resulting viral load for each of these species across all of these NEON sites, which is really cool because you can see very clearly sort of that difference in viral load that each species carries. And you can do that again from using NEON specimens. This microbiome study was an interesting use case, I thought. So in the aquatic macroinvertebrate protocol, we go out, we scoop water out of lakes or rivers or streams, filter the macroinvertebrates out, and then store them as bulk specimens. And this is what that looks like. But these researchers took these samples that we had, sorted them out into these insect orders, right? So we've got beetles, flies, mayflies, these insect orders, and then you use metabarcoding to characterize the gut microbiome for each of these orders. So what microbial associations might be important for each of these hosts. Trait and functional ecology is also pretty common. So this researcher took some pinned ground beetle specimens, measured morphometric traits from each of these pinned beetles, and then used them to test how each of these traits are evolving functionally and what ecological constraints might come into play for North American beetles. In the case of environmental processes, this researcher used our air-dried soil samples to characterize organic matter in the soils across each of these sites, right? So then they used sort of each of these fractions to test for drivers of that organic matter within that ecosystem. More recently, we've been getting a lot of requests for specimen images, mostly for use in developing AI tools, and mostly again for use in classification, which is pretty neat. And so these researchers took our bulk beetle samples, which, if you remembered, are not identified as species. It's just a jar of beetles, and developed some classification models to try and identify the species that were within those samples. And lastly, our specimens and data are used in education, outreach, and training as well. So a really nice example of this is, since we do put so much time into characterizing our mammal specimens, we obviously have a lot of downstream data for them. And so researchers used this data to create an online research or online resource for small mammal identification called whiskerwiki.org. And so a lot of the images and the data that we've collected are available through this tool and link back to our pages as well. With all that being said, we generally do a whole lot in regards to specimens and samples for the NEON project, and we're always definitely willing to talk to anyone about sample loans, or if you'd like a tour, or if you need help searching for samples or understanding what it is that we have. I think Callie already linked the contact us page, but this is the QR code to that contact us form that is available on the NEON site in case any of you need to get in contact with any one of us. We definitely reply pretty quickly. And yeah, thanks. And I will take any questions if anybody has any.
Thank you so much, Chandra, for covering all of that. And we'll wait just a minute. If you have a question, you can put it in the Q&A or in the chat itself. And while we wait, could you just maybe touch a little bit on, you mentioned destructive sampling, and we are trying to get people to use the samples. So is there any different processes that people follow if they're going to use it for destructive purposes versus regular request for a loan?
No, it just in the loans process, we do again ask, we do try to ask like what it is that you're using these for. But just because you're destructively sampling something does not mean you have sort of a less of a chance of getting those samples. That's not the case at all. Now, if of course you're destructively sampling again an entire sample type that we have, well, that's a little bit of a different story. But like there's no, oh, this person is destructively sampling this. Maybe we don't want to send that out. That's not the case. We do want people to use our samples, and there's no different protocol or way of requesting those samples that's different from someone who just wants to image them, for example.
Is there a
Just to
minimum
touch
request?
home a little bit on that part though, we do engage with the researcher early on to figure out their research design and the sample types, because different researchers' requests are different depending on their research question, how many replication, how many different sites, and all sorts of things. So we try to engage. We do have certain samples that can be destructively used that are in limited quantity. So we do want to kind of let the researchers know about the data types from our point. And usually with these discussions and meetings, we come up with a study design and then kind of loan process that satisfies both us and the researchers.
And as curators and from your team, do you see opportunities that maybe researchers would really want to think about exploring with the collections, given that we have extended collections as well?
Like Hojun, did you want to talk about the dark taxa?
Yeah, I mean, so we have a lot of samples, and then some of them are bulk samples, and those are, of course, invertebrate bycatch samples, but then there are aquatic samples and other bulk taxa. And to me, they are dark, meaning that they are not readily accessible in terms of species ID or number of specimens, but that's the source of information for biodiversity. If you want to really understand long-term ecological changes, those are really a cool source of information, and we do encourage people to think about these various ways. And although the NEON is developed for sort of ecological studies, as you saw in Chandra's talk, a lot of users and researchers are coming from many different disciplines. So we try to promote NEON Biorepository not only for ecology, but for all science. So anybody who's interested in using our samples, please do contact us.
And the contact us form is a great way to start at any stage of your project and have conversations with the experts. And is there a minimum sample size people need to request?
Nope.
minimum sample size. Perfect. Yeah. And if
You're
it
like, I've
can,
had people contact me to just be like, hey, can we please have a picture of this one beetle? And I'm like, okay, sure, absolutely. We'll get you a picture of that one beetle. And that happens. So it doesn't, nope. And I said, anybody, you don't have to be a researcher. You don't have to have funding. Anybody can do it.
great. Yeah. And if it turns out one of the samples that is popping up in the sample search is at a different repository, does the NEON Biorepository help coordinate that request, or should they reach out directly to that storage?
I mean, we can. Of course, we have the contact information for, all of those external locations. But of course, each of those locations has a different way of doing loans and processes that don't necessarily like constrain that aren't constrained by like our processes. So they do, whoever is requesting samples does have to go through whatever their rules are. But like I said, we can certainly help when it comes to at least contacting whoever needs to get contacted and who would know where those samples are and how to get those out.
Perfect. And I will leave just a few minutes here. If you do have a question, you can put it in the chat or the Q&A. Otherwise, the contact us form is a great way to get in touch in the future. Again, this recording will live on our science seminar page and on our website. So if you need to reference it in the future, you can find it there. It will also be on our Neon Science YouTube, which is a great resource for learning more about neon itself.